Hi, students. Today I want to give a lecture on this first part of the workshop, and basically this lecture involves an overview of chemotherapeutics. I'm going to give an overview of chemotherapeutics. Basically, the word chemotherapeutics refers to drugs. Firstly, let me talk about the outline of today's lecture. Firstly, I'll talk about the principles of microbial therapy and the general mechanism of actions, which is very important to understand how these drugs actually operate in the human body. And also of importance is the spectrum of activity, whether they are broad or narrow. And thirdly is drug resistance, which is a very important component because microorganisms have the capacity to develop drug resistance when they're exposed to excessive concentrations and excessive treatments of drugs. And especially nowadays when most of these important therapies for vital diseases such as HIV-related disease and TB, where they have developed drug resistance. It's important to know exactly how to avoid that, because if all drugs develop resistance, it will be difficult now to find new drugs that will be able to wipe out the infections. Lastly, I'll talk about an overview of antiparasitics, antimalarial, and antiviral drugs. Okay, firstly, let me talk about the transmission of infection. As you all know, I believe each and every one of you have been infected with some sort of pathogenic infection at any time during our growth stage. But it's important to understand that transmission of disease usually requires an unbroken chain of events. Usually, these are cyclic events, which we'll talk about later. And the major player in this cyclic of events is the pathogen, also called microorganisms. These microorganisms are infectious microorganisms, or what we call deleterious microorganisms. They are called pathogens. These pathogens normally must live and reproduce in what we call a reservoir. A reservoir is like a storage, structure. Such reservoirs can either be in a human being, an animal, or sometimes in soil. And for example, is a pathogen called influenza virus. Influenza virus, which is commonly known as flu virus, is an organism that reproduces in human being. So that's its reservoir. Having said that, let's quickly look at the chain of transmission of these microorganisms. On the left, you see that there's a diagram showing chains which are interlinked, representing the various stages of microbial transmission. And these have to be linked in the sense that they're not supposed to be broken. For the microorganism to survive, this chain of events must be linked at all times. Firstly, you have what we call a reservoir. As I said already, a reservoir could be a human being, an animal, insect, or soil. Basically, these reservoirs, they will store these cells of pathogen organisms. But for these organisms to execute a new infection, they must come out of this reservoir and use what we call a port of exit. Port of exit basically is a point, or is a system where these microorganisms leave the reservoir. For example, in human beings can be through nasal mucosa or oral mucosa. Yes, and once they leave the port of exit, which means they need to immediately, they must be able to travel to the next point there must be a mode of transmission. Mode of transmission can be either, for example, insect bite, nasal droplets. For example, if someone is sneezing without protecting their nose, and the droplets will just be flying all over, and susceptible people might catch those droplets, which are loaded with infectious, microorganisms. Yes, and that's how the transmission will happen. And sometimes it can be transmitted through semen. So once the transmission has occurred and landed on the next, what we call next port of entry, there must be an entry point. For example, in a human being, there are specific entry points in the human body which allows microorganisms to find their way inside the body. For example, the nasal mucosa, which is the nose openings, and those the walls of the nose inside is a mucosa, which means the tissues in those the nose is such that they are highly susceptible to infection. And once the microorganisms find themselves in the nose mucosa, they are specific receptors where they bind and find entry into the body. Or it can be oral mucosa through the mouth, or skin abrasion, where skin is basically opened by way of an injury, or it can be skin rupture. So these are some of the port of entry on a human body. What is also important, that such a human body must be what we call susceptible victim. What does this mean? That person must be in a condition, a susceptible victim must be in a certain condition. For example, a malnourished individual, or an immunized individual, or immunocompromised individual. A susceptible victim is a victim that lacks resistance against such pathogens. A human body is designed to resist infection under normal circumstances. But for a susceptible victim, who probably by way of malnutrition or unimmunized, gets into contact with such pathogens, may fall victim, because these pathogens basically, they'll find themselves inside and begin to multiply. And such a person becomes a reservoir, because they'll multiply to a high level and cause sickness and disease. So basically, looking at the chain of infection, on the right you have a diagram that shows various types of microorganisms that are likely to cause disease. You can have bacteria, virus, fungi, or parasites. And these, once they leave the reservoir, can be, for example, human being, water, air, medical equipment, etc. There are many sources of reservoirs. And yes, find themselves out of the body via a port of exit. As we said earlier, it can either be secretions, nasal mucosa, oral mucosa, or excretions. Yeah, and eventually find themselves in some form of a transport system, either through contact, directly or indirectly. You find themselves in the air, for example, when someone sneezes, those fine droplets, they are carried by the wind, and they carry to long distances and may land on susceptible human beings. Or it can be through a vector. A vector is another organism, for example, mosquito, or an insect bite that may, collect the blood or carry some of those microorganisms in its body system, and as it flies around, or as it flies to the next victim, it will transport that pathogen. Again, on the next victim, there must be a suitable port of entry, such as the mucosal lining or open wound or respiratory tract. And eventually, once they find themselves in, then it will depend on the condition of the individual. If that individual is a susceptible host or victim, he may succumb to that infection. Okay, so now, how do we control infection of diseases? Infection of disease can be controlled by breaking that chain of transmission. There are a couple of ways in which we can break the chain of transmission. The first one is by blocking the port of exit. You remember we spoke about the port of exit. Once you block the port of exit, the microorganisms won't be able to find themselves out of the body, out of the reservoir. And the second is destroying the reservoir itself. Okay, you cannot destroy the human being, but there are certain reservoirs that you can actually be able to destroy. For example, you can be able to destroy, water, for example, or insects that may be able to carry such. Yeah, and the third part is the third way of breaking this chain is by blocking the mode of transmission. For example, if you wear a mask, if you suffer from flu and wear a mask over your mouth and nose, such that when you sneeze or cough, those microorganisms will basically be confined to the mask, and that will block their transmission. And for example, if someone is suffering from venereal diseases, and one way of blocking the mode of transmission is by wearing a condom. So that way the microorganisms remain in the victim. Also by blocking port of entry. By blocking the port of entry, you'll be able to avoid the infection and colonization of the human being. The last method is by reducing victims' susceptibility. Okay, so reducing the victim's susceptibility can be done, for example, by vaccination. There are immune vaccinations, especially, for example, if those vaccinations that are given for under 5 vaccination for under 5 children, vaccination against viral diseases such as rubella, polio, and such disease. Or you can also, if the victim has optimal rest, it reduces the level of susceptibility. Yeah, and optimal nutrition. Optimal nutrition also is another way of reducing victim susceptibility. Because with good nutrition, the immune system is highly, boosted and is sensitive to infection and be able to control and safeguard the patient. Okay, now having said that, let's look at the definition of chemotherapeutics. Yes, what is the definition of chemotherapeutics? Basically, chemotherapeutics can be defined as drugs that eradicate pathogenic organisms or neoplastic cells in the treatment of infectious diseases or cancer. So these are basically drugs. They kill microorganisms or can eliminate neoplastic cells. Neoplastic cells, basically, these are cells which have gone basic during cancer development. They lose control and they grow without control by the body. They are not regulated, and there are such drugs which actually can eliminate cancer cells. Excuse me. So now let us look at the principles, how they exhibit, this chemotherapeutics. What are the principles of drug treatment? This basically, when you talk about principles, this brings us to this what we call selective toxicity. These drugs, they show or exhibit what we call selective toxicity, in the sense that Once a chemotherapeutic agent or drug is administered to an individual, that chemical will enter the body and move to the site of infection. And interestingly, the drugs are designed such that once they reach the site of infection, they will be able to destroy the pathogenic organism or the microbe without, and be able to eliminate it without actually affecting the structure and function of human body cells. This is a very important concept for you to understand. It's a principle of all drugs. And this is why when we take drugs, we don't die. Unless in certain situations where an individual is sensitive to certain drugs and they become, and experience adverse effects. Okay. So, before we move further, it's also important to understand the classification of various chemotherapeutic drugs. And once you understand how they are classified, then you understand how their mode of action are designed, because this is how they are classified. You have the first group, which is called antimicrobial drugs. Antimicrobial drugs are subdivided into 4 categories. The first one is what we call antibacterial drugs, which basically eradicate bacterial infections, bacteria microorganisms. The second is what we call antimycobacterial. Antimycobacterial are special drugs that target mycobacteria, microorganisms. As you know that there's a difference between bacteria and mycobacteria. Mycobacteria is a certain type of bacteria which has extra features. It grows like fungi, but it's bacteria. And it is characteristic of a pathogen that causes very serious diseases such as TB. Yes, TB is caused by mycobacteria, but we'll talk about this more later on. And the third group is what we call antifungal. These are drugs that actually eradicate fungal pathogens. The last one, which is category D, is antiviral drugs, which eliminate virus. Okay, so this is the first group, which is antimicrobial. Basically, it targets microbes. The second one is what we call antiparasitic group. So microbes are different from parasites in a number of ways. As you know, parasites are organisms that cannot thrive on their own, but they live on another organisms. These are, for example, protozoas and helminthids. So we have the first group, which is antiprotozoa drugs and anti-helminth. The last category is what we call anti-neoplastic drugs. As I said earlier, neoplastic drugs basically are drugs which target cancer cells. Okay? Now, let's go into details and look at the mechanism of action of antimicrobial drugs. What are the mode of action of such drugs? Okay? And so when we look at the mode of action, there are 4 categories by which such drugs actually work. The first category is where these are drugs which target the cell wall synthesis. So they work by inhibiting the cell wall synthesis. And as you know, the cell wall is an important characteristic of bacteria, for example. And by targeting those cell walls are degraded, they kill the bacteria. The second category is these are drugs basically that disrupt the microbial cell membrane. So it's important to understand, for example, bacteria has the bacterial cell covering has got, in the outer layer there's a cell wall, and inside the layer is what we call cell membrane, which is different from human cells. because human cells, they only have cell membrane, they do not have cell wall. So they are specific drugs that are designed to target such structures within the microbes. The third category is what we call drugs that inhibit protein synthesis. As you know, that protein synthesis is an important, biochemical process in every living organisms. So by targeting these pathways that synthesize these proteins, by inhibiting those processes, you actually eliminate such organisms. And the last one is by interfering with the metabolic processes. Metabolic processes are pathways that actually metabolize, for example, certain foods within the cells. Okay. Now, so basically, if you look at this diagram, basically it shows where these categories of drugs operate, where they target. You can see the arrows indicating, for example, the interference of the metabolic process which happens within the cell, in the metabolic process of folic acid, by interrupting the pathway where the arrow shows, that will block the formation of folic acid. As you know, folic acid is an important element. In fact, folic acid is a vitamin, and folic acid is also needed for the synthesis of nucleotides and amino acids. So by blocking the production of folic acid, actually, you are advantageously blocking the DNA and RNA processes. Okay, then you can see other points of action where the arrows are indicated. Okay, let's now quickly look at how antimicrobial drugs that inhibit cell wall synthesis. How does that happen? Okay, so it's important to understand that the cell wall is made up of two chains, what I can call a chain of proteins. These are two layers. These are two layers which are cross-linked by certain protein residues, which these residues basically they create some form of a protective structure to form what we call a cell wall. Under normal circumstances, the process of from 1 to 3 occurs in this cell wall synthesis. So what happens is that there's what we call penicillin binding protein. This penicillin binding protein basically is an enzyme called transpeptidase. Transpeptidase is an enzyme that catalyzes the cross-linkings of subunits from these cell wall chains. When they are cross-linked, so that they are actually intact and they form a strong structure for protecting the microbacteria. And what drugs, what such drugs that target cell walls have been designed to do is basically to target the transpeptidase enzyme, what we call penicillin binding protein. What they will do basically is to bind to this enzyme, and by doing so, basically they disrupt what we call the beta-lactam ring. When this beta-lactam ring opens, it makes the penicillin binding protein, it actually disables the penicillin binding protein. By disabling the penicillin binding protein, it will not be able to form those cross-links in the cell wall. And so there will be disruption of cell wall synthesis. And any bacteria that hasn't got a cell wall basically will die, because that's an important protective structure. And some of the examples of such drugs are penicillins, cephalosporins, and vancomycin. Basically, these are drugs that target cell wall synthesis. And the second group of drugs are drugs that disrupt the microbial cell membrane, which is an inner membrane inner to the cell wall. And as you know, cell membrane is a lipid bilayer, and within those cell layers, for example, in bacteria, you've got what we call proteins that are called ergosterol. Ergosterol proteins are membrane proteins. They are not transmembrane proteins, they are membrane proteins, which means they are located within one layer of the protein. So how does that cell membrane disruption occur? For example, a drug called amphotericin B, once administered, it goes to the cell membrane, and once it reaches cell membrane, it targets ergosterol protein. It interacts with those proteins. By interacting with those proteins, basically, it breaks down the proteins to create a pore, an opening within the cell membrane. And once that pore is created, the macromolecules, the small molecules and ions will begin to leave the cell. For example, important ions such as calcium and sodium and potassium will start floating around, will leak out of the cell, and that will disrupt the function of the bacteria or microorganisms, and eventually it will die. Okay, let's now look at how drugs inhibit protein synthesis. So here basically we have two mechanisms that are at play. How they inhibit protein synthesis. The first one is the protein inhibition can occur at the site of protein synthesis. As you know, at the site of protein synthesis, there are important, molecules which are called ribosomes, which are involved in the protein synthesis, and these ribosomes usually occur in what we call pairs. They are called ribosomes subunits. We'll look at this later in detail and how these basically are targeted. And the second mechanisms is when basically these drugs enter the nucleus and prevent the synthesis of nucleic acids. And when nucleic acids are prevented, they block the transcription, which is the multiplication of DNA, and the RNA synthesis. Okay, so for such kind of drugs, we have three examples. For example, tetracycline, gentamicin, and erythromycin operate by inhibiting protein synthesis. Okay, now let's move on to the next category of drugs. These are drugs that interfere with the metabolic processes. As you know, metabolic processes, basically, these are processes that metabolize foods, and for example, they degrade foods to smaller particles so that the elements can be used for biochemical synthesis and such things. So with such kind of microbial agents, basically they inhibit the synthesis of folic acid, which is a very key process in the microbial biosynthesis pathway. So what basically happens is that these drugs, they will target, they will compete with what we call PAB. Basically, is a benzoic acid. It's a 4 benzoic acid molecule. What they'll do is they'll bind and incorporate themselves into the structure of folic acid and interfere with the metabolic process. And eventually there will be failure of synthesis of folic acid. And we have 2 examples of such drugs which interfere metabolic process. We have what we call sulfonamides and trimethoprim. These 2 examples are drugs that interfere with metabolic processes and thereby killing the microorganisms. So here is a diagram basically that shows the various sites where such drugs, interact with the cell. As you can see on the left there, we have this. On top, the outer layer of the organism is what we call cell wall. At cell wall level, you have drugs that actually inhibit cell wall synthesis, such as D-cycloserine, vancomycin, bastracin, penicillin, cephalosporin, and cefamycins. So these are a group of drugs that target the cell wall synthesis. And there are also some drugs that target the cell wall integrity. By disturbing the cell wall integrity, they'll actually disrupt the cell wall, such as beta-lactamases. So this is just one of the examples. And of course, if you look at the diagram, you'll see, for example, the various drugs, examples of drugs that operate at various sites. For example, DNA synthesis, that block DNA gyrase, RNA polymerase, and the drugs also that target the cytoplasmic membrane, or translation of proteins, or synthesis of proteins. So basically what you need to do with such kind of a diagram is just understand the sites where this inhibition occurs and how it occurs. Importantly, I spoke about protein synthesis. Protein synthesis basically is inhibited by targeting what we call ribosome subunits. It's important to understand that even in human cells you have ribosome units, subunits rather, but the the type of subunits found in human cells are different from subunits that are found in microorganisms. Here we have 5S subunits which work in pair with 30S subunits. But in human beings we have, for example, 80S, or sometimes we have 60S subunits which work hand in hand with 40S, So the combination of these subunits are unique between the human cells and bacterial cells. And also, it's important to understand these 2 concepts: bactericidal action versus bacteriostatic action. So, drugs can react with, can eliminate microorganisms either by what we call bactericidal action. Bactericidal action is a drug action that leads to the death of the susceptible microbe. For example, through inhibition of cell membrane, such as penicillin. So it's important to understand this. This bactericidal is like suicidal. So basically they kill the microorganisms. Whereas bacteriostatic action is a drug action that inhibits the growth and the spread of the microbial population. So it's important to understand this, that these microbes basically they do not kill the bacteria, but they simply inhibit the growth and spread. Okay, let's quickly look at the various activity, the spectrum of activity. What is the spectrum of activity of any drug? Basically, the spectrum of activity of a drug, tells us how many types of microbial species that each drug is able to kill or eliminate. How many microbes are sensitive to that such particular drug? This is what we call spectrum of activity. So when we talk about spectrum of activity, we can talk about broad spectrum or narrow spectrum. So we'll talk more about this later, but before we talk about that, it's important to understand various concepts here. Concepts that will help us, describe the various spectrum of activity. It's important to know that bacteria are classified, for example, according to their diagnostic, methods. How do you detect bacteria? There are, for example, two methods that are used to detect bacteria, and this is how they are classified. Firstly is by gram staining. Gram staining is the diagnostic test, lab test, which when you stain, it can either give you a positive result or negative result. And by doing so, it will basically tell you what type of organism. But not only that, because that on its own is not informative, but it has to go hand in hand with the detection of bacterial morphology. The bacterial morphology is the shape of bacteria. So here are basically groups that have been identified based on the classification of bacteria based on the staining and the morphology. There are basically 8 groups. The first group is what we call gram-positive cocci. So you can see gram-positive cocci is a type of bacteria that stains positive with the gram staining and has cocci, which is the form of morphology, shape, and which is different from the second group, which is gram-negative cocci. The third group is called gram-positive bacilli. The fourth group is called gram-negative rods. So you can see that the morphology here basically will distinguish between the positive and the negative. Rods are like very stretched rods. Bacilli are like round structures which are connected together. Also, you can also have what we call anaerobic organisms, which are these are microorganisms that grow in environments without oxygen, anaerobic environment. You can also have what we call spirochetes species, which can cause, for example, leptospirosis, and which basically can cause trypanosomes, trypanomatosis, such as syphilis, which are sexually transmitted diseases. These are also a different group of microorganisms. And this followed by mycoplasma species. Mycoplasma species basically are microorganisms that they cause, for example, pneumonia. They have typical, shape and typical, characteristics. As you can see, myco there basically means they have some form of, fungal characteristics, but yet they are bacteria. And the last group is what we call chlamydia. Yes, chlamydia basically are microorganisms that cause STIs. For example, sexually transmitted diseases such as gonorrhea in human beings. Okay, it's also important to know that there are other minor groups which are not characterized among these 8 groups. They are also included, as important, groups. So, based on those morphology and based on the characterization of these microorganisms, scientists have been able to determine the spectrum of activity of such microorganisms. The first group is what we call narrow spectrum, microorganisms or activity. These are drugs basically which act on a single or limited group of microorganisms. They are highly specific. For example, isoniazid, which actually eradicates TB. They react on TB alone. They cannot react with any other microorganisms. And then we have the second group, which is slightly extended spectrum. Extended spectrum in the sense that they are not very broad, but within they are able to, for example, work effectively against gram-positive and gram-negative bacteria. They are slightly broader spectrum than narrow spectrum, although they are not that broad. So they are somewhere in between. For example, ampicillin. And the last group is what we call broad spectrum, drugs or broad spectrum activity. These are effective against a huge variety of microbial species. For example, tetracyclines are against gram-negative rods, anaerobic organisms, spirochetes, mycoplasmas, chlamydia, amoeba. Almost all those groups that we discussed earlier can be controlled by broad spectrum drugs. One important feature about broad spectrum drugs is that these drugs are susceptible. They are not suitable because they are likely to develop drug resistance because of their broad spectrum nature. So they don't target one microorganisms, they may target other organisms. And in so doing, they will limit. In so doing, they will limit the production, the reaction. Okay, there was a noise here. I just want to pause my recording and check what is happening because it looks like there's a problem. So I'll continue this discussion later. Okay, let me continue and see how far we will go basically. Yeah. Now let's look at microbial resistance of drugs. It's a very important concept in the sense that if not properly managed, administered, drugs can develop resistance to microorganisms. And as you know, when microorganisms develop drug resistance, what it means is that they cannot be controlled by any drugs. And that will lead to vicious infections and may lead to death of individuals. Now, there are various ways by which microorganisms can develop drug resistance. The first one is when such organisms produce enzymes that actually destroy the active drugs, which means that microorganisms, they are able to counter these drugs by producing specific enzymes that destroy the drugs. And the second one is microorganisms that are able to change the permeability to drugs. So they might probably develop structures that will prevent drug permeability into the cell. The third group are microorganisms that develop altered structure targets. You remember we talked about structure targets, drug targets on microorganism cells, such as receptors and other structures. So there are certain organisms that are able to change the shape or to change the form of these receptors to elude the drugs, which means when drugs are administered, they won't be able to bind to these receptors. and that way they'll develop resistance. The fourth mechanism is when actually these microorganisms change the metabolic pathway so that they are able to bypass the reduction inhibition of the drugs. Yes, and this is really possible by changing the metabolic pathway. The last group of mechanism is what we call when microorganisms basically alter the enzymes that can still perform in metabolic activity. So if an enzyme is targeted by a drug, an organism may actually change the formation of that enzyme such that when the drug is administered, it won't be able to recognize the active site of the enzyme. And that way, the enzyme will continue to perform its metabolic activity. Okay. Let's move on to cell wall inhibitors. Cell wall inhibitors. So firstly, on cell wall inhibitors, remember we spoke about penicillins as one of the drugs that target cell wall inhibitors. What are penicillins? So basically, penicillins are a group of drugs, and these drugs basically are basically designed to target cell wall, disrupt cell wall synthesis. There are different types. The prototype is what is called penicillin G. The other name for such drug is benzylpenicillin. Out of this prototype, many have been derived. These drugs have what we call long-acting, activity when they are administered intramuscularly. So they target, they are administered intramuscularly. So you should know where these drugs are deposited when they are administered intramuscularly, because we spoke about this in the previous lecture. Also, we have what you call beta-lactamase resistant penicillins, such as flacloxacillins. These are effective against beta-lactamases. And within the same group, we also have what they call broad-spectrum penicillins, such as amoxicillin. These are effective against numerous species of gram-negative bacteria. So penicillin is a collection of a large number of of drugs which have these three types of actions. We also have what we call clavulanic acid and tazobactams. These drugs, when are combined, actually, they are combined with penicillins in order to prevent drug resistance. As you know that penicillins you have penicillins which are broad spectrum, and in order to prevent drug resistance development, you can actually combine penicillin with either clavulanic acid or tazobactams, and that will prevent the inactivation of such drugs. When you do such kind of combination, it's important to consider the clinical, implications. For example, some patients actually can develop allergies. It's been shown that 10% of clients may develop allergies as a result of such combination. So you need to be aware about that. Also, you should be notiful about the renal or hepatic function of such patients. Because, for example, if the kidney and the liver are not functioning properly, the metabolism of such drugs may be difficult and may end up actually getting low drug activity. Okay, the next group is what we call cephalosporins. Again, cephalosporins are these groups of drugs that target the cell synthesis by inhibiting cell synthesis. These are drugs basically have a wide, spectrum against gram-negative bacteria. They are more wider than penicillins. And when you administer cephalosporins, it's important that you look at the history of clients. If the client has got, has experienced of, for example, allergies, for example, such as urticaria or anaphylactic, reactions. These are very strong and serious allergic reactions due to such kind of drugs. You need to avoid, administration or prescription of these drugs to such patients. So, in such a case, you might want to use penicillin, or carbapenems. Okay. Also, there are other beta-lactam, drugs. For example, carbapenems and monobactams, which can also be used, to control microorganisms by inhibiting cell synthesis, cell wall synthesis. Now, the advantage of such drugs basically is that they have activity against bacteria that becomes resistant to penicillin and aminoglycosides. So when you encounter a patient who is or if you encounter bacteria that is resistant to penicillins and aminoglycosides, you can actually use beta-lactams as an alternative. The important feature about these kind of drugs, basically they are administered parenteral only, which means via injection. Okay, there are still more groups of drugs. For example, polypeptides and the glycopeptides antimicrobial agents. As you can see, polypeptides, basically these are drugs that formulated from a group of peptides, whereas glycopeptides, these are proteins which have got sugars on them, and they are used as antimicrobial agents. Usually such antimicrobial agents are relatively narrow spectrum agents, which means they have action against few bacterial species or microorganisms. Are usually effective against gram-positive cocci. An example is vancomycin and the teicoplanin, which are important in the treatment of MRSA, which is a methicillin-resistant Staphylococcus aureus. These are Staphylococcus aureus, which is resistant against methicillin drug. In such kind of cases, you can use vancomycin and teicoplanin. Now, the other important feature about such drugs is that their use usually is reserved until less toxic antimicrobial agents have been tried and when there are no alternative drugs available. So this is one of the important considerations, because you don't want to use such drugs which are very important features. For example, they might excuse me. Yes. Okay, let's move on to the next group of agents that inhibit protein synthesis. The examples here are tetracycline, as we saw earlier. Among the tetracycline, you have examples such as the doxycycline and the minocyclines. The second group is aminoglycosides. We have gentamicin, streptomycin, neomycin, and tobramycin as examples of aminoglycosides. Macrolides is another group of drugs. Some examples of macrolides as azithromycin, erythromycin, and yes. Those are the two examples. And then also you have chloramphenicol, clindamycin, streptoglamins. Yes, these are 6 groups of antimicrobial drugs that are able to inhibit protein synthesis. So just if you can memorize 2 and give examples of those 2 or one, that would be interesting. And let's look at tetracycline. Tetracycline is a broad spectrum, antibacterial, which has bacteriostatic action. Remember, we spoke about bacteriostatic action. Tetracyclines are highly used, especially in sub-Saharan Africa, because they are cheap and also that they are broad spectrum. Yes. The common adverse effects include gastrointestinal disturbances. Some patients can develop hypersensitive reactions or what we call superinfection. So superinfection is where a new infection emerges as a result of killing the first infection, the first bacteria, the first line of bacteria. So because of such broad spectrum bacteria, they might kill other flora and changing the chemical environment, for example, of the GI. And that change in chemical environment may lead to a new infection, and that's what we call superinfection. Okay. So such drugs basically they bind to free divalent or trivalent ions, especially calcium. Remember, ions are important, for the survival of microorganisms. They are involved in a number of biochemical reactions. And with such drugs, tetracycline, they are not recommended for children under 8 or to women who are in latter or half of the pregnancy, because of they affect bone development. As you know that they bind to calcium, which is very important for bone formation, especially in children under 8 years, or women who are pregnant. They might affect the fetus bone development. So you need to be very careful when administering such drugs. Let's look at aminoglycosides. Basically, these are drugs which are effective against a wide range of gram-negative bacteria. For example, Staphylococcus and the Streptococcus, streptococci, microorganisms. And one important characteristic is such drugs are administered only via injection, usually once daily dose, up to 12 hours for specific conditions. Because one of the reasons is they cannot be administered through the GI is they can be degraded by the enzymes, or the pH might interrupt the activity of these drugs. Then the next group of drugs is called macrolides. These are basically drugs that specifically function by attaching themselves to the large ribosomal subunits. And when they do that, basically they inhibit protein synthesis. Also, because they are a spectrum of activity, they are important alternatives for clients with an allergy or penicillin or other related antibacterials. So in a situation where a client has got allergies to other drugs, this is a good alternative. Let's look at streptogramins. And streptogramins, basically, we have these are drugs basically that act by binding to 50S ribosome subunits, and when they do that, they prevent the elongation of the peptide chain formation, causing incomplete chains, for example, to be released. And those incomplete chains basically are non-functional chains. So they become useless and they cannot be used in the synthesis or in the well-being of microorganisms. They become non-functional proteins. An example is quinaspiritin or dalphospritin. These are good examples of streptogramins. Usually they are indicated for intravenous administration in patients that, or in cases where there is MRSA or vancomycin resistance, where there's vancomycin resistance suspected. Chloramphenicol, another very common antibiotic or antimicrobial drug used in sub-Saharan Africa. It's a broad-spectrum drug and is effective against gram-positive and gram-negative bacteria, Rickettsia, chlamydia, and Coxiella. So you can see there's quite a broad spectrum activity. But interestingly is that such drugs basically have toxic effects to mammalian cells, especially bone marrow cells. So caution needs to be exercised when administering such drugs. And usually, administration of such drugs is restricted to severe infection, where you have a client with severe infection that are likely to cause death. Okay. Then let us look at drugs that basically inhibit cell membrane. Cell membrane, basically they target the cell membrane and disrupt the cell membrane, creating pores, and these pores basically release the ions from the inside cells and disrupting the function of cells and killing the cells. An example is polymyxins. Polymyxins basically are drugs that are derived from Streptomyces moths. These are like yeast moths. And this is where they derive such a drug. And a good example also are colistin and the polymyxin B. And when administering such drugs, it's important that you monitor the kidney function, because if the kidney is not functioning properly, they might end up accumulating in the body, forming toxic levels. Next are quinolones. These quinolones basically are known to be folic acid antagonists. Remember, folic acid is a vitamin B, and microbes always synthesize their own folic acid, and quinolones target such biosynthetic pathway. An example of quinolone is norfloxacin, ciprofloxacin, moxifloxacin, and as these are some of the examples. Basically what they do is they inhibit bacterial growth by preventing DNA synthesis prior to mitosis. So, and also these drugs are active against a range of gram-negative and some gram-positive bacteria. So they're quite effective, drugs. Let's look now at tuberculosis. Tuberculosis, one of the important diseases of sub-Saharan Africa, including South Africa. It's abbreviated as TB. TB is caused by mycobacteria tuberculosis. This is a microorganism or pathogen that is likely to infect a large number of populations, especially in environments where there are social inequity or overcrowding of people. For example, in informal settlements, in townships where the population is large. In such kind of environments, mycobacterial tuberculosis actually thrives very well. What are the TB drugs or TB agents available effectively to eradicate TB? TB is a worldwide disease that affects lungs and lymph nodes, and it kills approximately 3 million people per year. So, and that is quite very, very huge population. The first line of TB treatment are as follows: ethambutol, isoniazid, rifampin, and pyrazinamide. So these are the first line of anti-TB drugs. By first line meaning, before you try any other drugs, you need to try these drugs to clear the TB infection. One of the important features of TB treatment is resistance. As you know that mycobacteria structurally are different from bacteria. They have a stronger cell wall structure, way much stronger than the bacteria. And this requires a specific drug therapy. And this is a therapy that is quite often a long-term therapy that degrades this cell wall, thick cell wall, at a steady rate over a long period of time until the microorganisms are cleared. Leprosy. Leprosy is also a disease that is caused by mycobacteria. It is a condition of skin infection or infection of the mucous membranes. Mucous membranes, for example, the nose, the mouth, or peripheral nerves can infect peripheral nerves, the nerve ends. Once it destroys the peripheral nerve ends, the patient basically loses the sensation of feeling. He's unable to feel. For example, when the hands come close to heat, the patient will not be able to feel the heat. If they touch the hot plate, they will not be able to feel the heat. As such, usually the patients end up burning themselves and losing their fingers. And also the nasal mucosa can be infected. Leprosy, just for interest's sake, the infection rates of leprosy in the world as at 2018 were 280,690 new cases of leprosy, which were identified in 127 countries. So it's still a huge problem and it needs to be controlled. Now, what are antileprotic drugs? The first one is called dapsone. Dapsone basically is a sulfone, is an antagonist, meaning it antagonizes the folic acid biosynthesis. And mainly bacteriostatic, which means basically that it does not kill the bacteria, but it inhibits its growth and spread. It may also have a slight bactericidal properties, slightly, not mainly. And quite often the treatment is long-term, sometimes for life. Yes. And other examples of such drugs are rimpicin, prednisolone, clofazimine. These are some of the antileprotic drugs, examples of antileprotic drugs. Okay, let's move on to protozoa infections, drugs that basically control protozoa infections. Protozoa infections, these are infections that are caused by very little organisms, for example, amoeba, flagellates, or sporozoites, or sporozoans. And the most common drugs are nitroamidazoles. There is a group of drugs. Yeah. And one example is metronidazole, which basically which actually after its action it is metabolized. Yes. By binding to the DNA and preventing transcription and replication of DNA. An example of antiprotozoal drugs is pentamidine and paramycin. Sorry. So those are two examples of antiprotozoal drugs. Okay. Now let's move on to worms. Worm infection. Also very interesting, although they are not very usually they don't cause serious problems, but usually these are quite widespread. For example, nematode infections called Ascaris, lumbricoids, or Enterobius vermicularis. So as you can see in the picture there is a picture of these worms. Sometimes they can be much smaller, very tiny white, worms that can actually infect the GI system. And quite often you notice that you have worm infection of those nature when you feel, your anus, something wiggling at your anus. You feel very discomfort with that wiggly, and usually you are tempted to rub or itch. By doing so, basically you are actually spreading the eggs. These worms come to the anal opening actually to lay eggs. It's a form of transmission of those eggs, especially when it involves children will literally use their fingers to scratch, and by doing so they'll actually collect the worm eggs and transmit them to other human beings. Okay, so you can also have what we call trematode infection. Okay, these are various types of worms. Sometimes you have what we call beef tapeworm. Cystode infections usually are found in water and can be infected with snails. They can infect snails, which form part of the parasitic life cycle. So if you have kids that like to play in water or small streams where there are a lot of snails, most likely they'll get such worms. Antihelmintics. Antihelmintics, basically these are drugs that are able to control certain parasites. For example, benzimidazoles. Benzimidazoles, they act intraintestinally and selectively disrupt tubulin formation. Okay, but quickly, tubulin formation. Tubulin are structural proteins, usually structural proteins in cells that help to form the shape of the cell. Also, they are important for mitosis process. So these drugs, what they do basically, they target those tubulins and disrupt those tubulins, and the cell basically loses the mitotic process, cannot multiply. Pyrantel, another drug which stimulates nicotinic receptors. Remember we spoke about nicotinic receptors, which basically are involved in regulating ion inflow and outflow. Levamisols are drugs which relate basically whose activity work on nicotinic receptor as agonists. Okay, let's move on quickly to antimalarial agents. Antimalarial agents basically are drugs that are designed to eradicate malaria parasites. For example, pyrimethamine, which basically antagonizes folic acid biosynthesis. They target the folic acid synthesis and disrupt that process in these parasites. Proguanil is another drug which inhibits pyrimidine synthesis and nucleic acid synthesis. Chloroquine, very, very common anti-malarial drug, especially in sub-Saharan Africa. Yes, these drugs are used as prophylaxis and treatment, which means they are used for prevention and treatment. Their mode of action is basically by suppressing the DNA replication and the transcription in the parasite. We also have a drug called quinine. Quinine is rarely used because of its adverse effects, but it acts on DNA by binding to DNA and prevents its replication and transcription. The last group is a drug called primaquine, which also acts on cellular structures such as mitochondria, where it suppresses the normal cellular metabolism. So these are some of the anti-malarial drugs. Let's move on to viruses, which is basically an important, disease agent. As you know, right now, the entire world is alert against coronavirus. As you know, there's an epidemic of coronavirus, which is started in Wuhan, China, and is spreading across the world. Viruses are very notorious, organisms that are capable of disrupting human life. The interesting part about viruses is that their ability to integrate their DNA molecule into human cells, and they use human cell mechanism to multiply. So there are a number of such drugs available. Well, not a number, so to say, but usually they are low, drugs that are able to control viruses. And usually this so basically most of these viral diseases can be prevented by such vaccine. For example, can prevent such can be used by controlling measles, mumps, rubella, polio, chickenpox, smallpox, hepatitis A and B, and rabies. As you know, these are most of the viruses which attack especially children. And by law, every child must be vaccinated against these, viral infections by 5 years. By 5 years, every child must have been vaccinated against these very deleterious viruses. Okay, so what are common antiviral drugs? So the common ones, for example, is aciclovir, also known as Zovirax, is important for controlling herpes infections. Amantadine, also called Chemotherapeutics, is important for influenza type A. Influenza, that is a flu. Didanosine, also called Videx, is important for control of advanced HIV infection. Ribavirin, also called Virozole, is important for the control of respiratory syncytial virus called pneumonia. Ganciclovir, also called Cytovin, is important for cytomegalovirus infections. For example, retinitis. For example, retinitis, this is a viral infection that infects retinal tissue. Retinal tissue basically is a tissue that is located in the retina. Retina is a part of the eye that is sensitive to light, which perceives light signal. And what will happen is that when viral infection occurs, there will be an inflammation of the retina, and that will disrupt the light perception. The last one is zidovudine, which is also called AZT. It's used as retroviral, It's used for retroviral infection in HIV. Okay, so while on antiviral drugs, there's a WHO recommended therapy of HIV and AIDS treatment. This is what is called highly active antiretroviral therapy. Highly active antiretroviral therapy basically is a therapy that consists of a combination of drugs. And the role of these drugs basically is to reduce the viral load and increase CD4 count. So these are immune elements. By increasing the CD4 count, it increases the immune system and prevents subsequent infections. And also it delays the onset of AIDS and prolongs survival of AIDS patients. Okay. So this HAART can be a combination of what we call nucleosides analogs and protease inhibitors. Remember, protease are enzymes that degrade certain proteins. Yeah. So with these protease inhibitors, once administered, they will prevent these proteases. They will inactivate proteases. Also, we have nucleoside analogs, which in such kind of a combination will actually improve the livelihood of AIDS patients. Okay, so that brings us to the end of this lecture, and I hope that you have actually learned how these various drug therapies operate. It's important because these drugs actually, this information is vital for your 83 assignment, because each group has been given 2 drugs to investigate, and you need to look up for information on various aspects of those drugs so that you know exactly how they their mode of action, the adverse effects, and various aspects about those drugs. This is a very important, exercise to give you a lot of knowledge and skills. Knowledge about these drugs and how they operate. Especially you become more conversant with pharmacological therapies, and also it gives you skills on research and group work. Okay, so before I end, it's important to remind you that next week we have a class test. Please get ready. Yeah, hard work always pays. Hard work will always pay. So please put in all your best. And I expect a lot of HDs from this class because you've been very, attentive and highly participative. So I like this class very much, and I'm enjoying your participation so far. You're very disciplined students, and please keep it up and work hard. If you have any questions regarding the test, please don't hesitate to ask me. Okay, by that now we come to the end of this presentation. Goodbye.